All right, so this is lipids, and for lipid, we say they don't form true polymers. Now, don't be confused. It's a large molecule, but the reason we say they don't form true polymers is because they are made up of different fragments of elements and not that. Now for carbohydrates, you can say they are made up of that the monomer for carbohydrates are monosaccharides. And so glucose and glucose uh can form maltose, glucose and galactose can form lactose, glucose and fructose can form sucrose. Now, but when you look at glucose and fructose, they are both monosaccharides. When you look at glucose and galactose, they are both monosaccharides. So when you look at what is the building block for all disaccharides, they are generally called monosaccharides. But for lipid, it's not like that. So you can have glycerol, which is extremely different, and you can have fatty acid. They are not of the same class, making up triglyceride. So those are not different, the same class of molecules. You cannot classify them in the same category. So in such case, we don't call them polymers because they are made up of entirely different fragments. All right. So we say they do not form true polymers. The unified lipid feature of lipid is having little or no affinity for water, so we call them hydrophobic substances, right? We say they are hydrophobic, which is what you see right here. They are hydrophobic, right? Now, and the reason for that is because they are mainly hydrocarbons, and that obviously form non-polar covalent bonds, whereas water is polar, and polar and non-polar substances do not mix. All right. And you remember the idea of water forming being polar compounds, right? Because of the electronegativity of oxygen in that relationship. Now, the most biologically important lipids are the fats, phospholipids, and steroids. And I'm going to touch base on them as we go. So fats, they are constructed from two types of smaller molecules, which are different. Like I told you, we cannot categorize glycerol and fatty acid into any category. They are extremely different categories. So we can't say they are monosaccharide, we can't say they are fatty acid, we can't say they are these. So they are extremely different, right? Now, so fatty acid is a group, glycerol is a different group. Unlike carbohydrate, you can say these are monosaccharides, right? Now, so when you look at glycerol, you see there are 3 carbon compounds there. So glycerol is a 3-carbon alcohol. So it's an alcohol, for instance, right? Then it is attached with, now, so it's this, then you see OH attached to each of them, which signify that it's alcohol. Now, then you see fatty acid, which here is fatty acid right here. All right, so this is carboxyl group here. These are carboxyl group there. Then it will be attached to the glycerol. All right, so that makes it triglyceride. So tri, 1, 2, 3, fatty acid, right? Then glycerol. So triacyl glycerol, meaning that 3 fatty acid glycerol. Now, so fats are separate from water because water molecules hydrogen bond to each other and excludes the fat. I told you about that already. It's because one is polar, whereas this is non-polar. In a fat, 3 fatty acids are joined to glycerol by an ester linkage. So the name of the bond is called ester linkage, or you could see it called glycosidic bond, depending on the resource you are looking at. All right, so it creates what we call triacyl glycerol or triglyceride. All right, meaning that they are very fatty acid. Now, here is fatty acid. They could vary in length and it could vary based on the number of double bonds that are there. Now, if you look at this, we say this is a saturated fatty acid, and then you have the unsaturated one. Now, when we say a fatty acid is saturated, what do we mean? Now, saturated simply means that, number one, it has single bonds in there. Now, if it is predominantly filled with single bonds, then it's going to be saturated with hydrogen atom. So that's the idea. So saturation is dependent on the number of single bond. So single bond means more hydrogen, saturation, densely packed. Then unsaturated means it has double bond, then less hydrogen. The more the bond, if it is all single bond, more hydrogen. Double bond, then less hydrogen. So you're going to see that in a moment as we go. Now, so if you look at this, for instance, now, I'm going to show this to you perhaps on the next slide. But when you look at this, this is saturated fat. This is what you get in any food that is from animal source. All right. So if you think about how your some meats that are fat looks like, you'll notice that at room temperature they become solidified. That's what this picture is telling you. So your butter that is from animal source is going to be solidified because it's saturated fatty acid. Those kind of fatty acid are difficult to digest. Unlike these guys, fatty acid that are unsaturated, that are from plant source, are unsaturated and they are easy to digest. That's why you notice that they don't solidify at room temperature. All right. Now, so fats made from saturated fatty acid are called saturated fats and are solid at room temperature. You see that? They are usually, now you see, most animal fats are usually saturated. They are saturated, and that means they are filled with single bonds. They are difficult to digest, and they are not as healthy. Now, fats made of unsaturated fatty acid are called unsaturated fats or oil, and they are usually liquid at room temperature. That's why you notice that your vegetable oil is always liquid. It's never going to solidify at any point, regardless of the temperature in your room. Now, plant fats and fish fat are usually unsaturated, and that makes them more healthy to eat. That's why it's advised that you eat vegetable oil rather than any form of oil. You notice that almond milk, soy milk, they are usually more healthy than the milk that come from cow. Anything like plant source, they are usually more healthy, they are easier to digest than what the animal source. All right, the major source of fat, the major function of fat is to give you energy. Somebody will, it look interesting to you, right? Now, there's it. Carbohydrate gives you energy, but fat gives you about 2.5% more energy than what carbohydrate gives you. The only challenge is this: plant fat will only supply you energy when carbohydrate is not available. As long as carbohydrate is available, you will not get energy from plants. So from fats are not readily available. They are not readily accessible. They only become accessible when carbohydrate become unavailable. All right. So fat is a compact way of animals to carry their energy stores within them. Now, in a phospholipid, two fatty acid and phosphate groups are attached to a glycerol. So this is what you see here. And that is actually what makes what we call the cell membrane. All right, so what we call the cell membrane, it has a phosphate head, which we call the phosphate head, and the fatty acid tails. All right, so the phospholipid means that there's a phosphate head and then the lipid fatty acid tail. All right, so there are two fatty acid tails, then there is a phosphate head right on there. Now, this doubled is what we call the cell membrane, and I'll show that to you as we go. Now, here is the cell membrane and how the orientation is oriented. So if you look at this now, this is a phosphate head, and each phosphate have two fatty acid attached to them. Then on this side as well, you see the phosphate and the fatty acid attached to them. Why are they arranged that way? They are arranged that way for adaptation mechanism, not to maintain, not for thickness, but for function. Now, on the exterior of the cell, you don't want the cell to, because the phosphate head is hydrophilic, so it's able to relate with water without fear. Whereas the fatty acid are hydrophobic. So the fatty acid that are hydrophobic, they cannot, water should not touch them. All right. Now, because of that, we need the phosphate edge to be on both sides, either in the inside of the cell because it's going to be exposed to water, or on the outside of the cell because it's going to be exposed to water. No side of the cell is shielded or prevented from water. Both sides must touch water. So what the phosphate does is to serve as adaptation mechanism, not primarily for thickness of the membrane, but first for the functionality as to prevent the trauma that happens between the water and the hydrophobic area of the membrane. All right. Now, so we have steroids, which are lipids that are characterized by carbon skeleton consisting of 4 fused carbon, which is what you can see here. Then you have cholesterol, which you see in animal membrane as well. All right. Now, let's go ahead and look at proteins. Now, proteins account for more than 50% of dry mass of most cells. And the major function of protein is the major function of the cell. All right. It's responsible for defense, for storage, for transportation. It helps with communication within cells. It helps with movement and structural support. All right. Now, here are proteins and how they function. So there are some proteins that function as enzyme. Some protein function for storage purposes. Some function for hormonal purpose, like hormones. Some functions in movement, contraction, that is contractile and motor protein. There we're going to be talking about actin and myosin. We'll talk about troponin and tropomyosin. Don't worry, for some of you that will take A and B, that will make sense to you later. Then we talk about defensive protein. We'll talk about the antibodies, transport proteins. You talk about the proteins that are responsible for transportation, aquaporins, glucose transporters, all of that. We talk about the receptor proteins that help to receive signals that is coming to a particular cell. We talk about structural protein that gives strength to the and resilience to the cell. We talk about collagen fiber when we get there. All right, so those are proteins and their type. Now, protein are formed by the monomer, which is called amino acid. All right. Now, the amino acid are monomers. When they are joined together, they have to be bonded together. The bond that joins them together is called peptide bond. And so when you see here, so the bond is called peptide bond. So when the chain continues, so multiple amino acids are joined together by peptide bond, then it is going to form a polypeptide chain. All right. When the chain is going to form a polypeptide chain. So you see polypeptide chain are unbranched polymers built from same set of 20 amino acids. Now, a protein is a biologically functional molecule. Now, the difference between polypeptide, which could be chain of amino acid connected by polypeptide bond, and an actual protein is that proteins are usually functional when they become three-dimensional in shape. When they become coiled, twisted on each other, that's the point when they are functional. I'm going to talk about that. So amino acid are organic molecules with, you see there's a carboxyl group here, which is carboxyl. There's a amino group here, and you see the R group, which is the side chain right there. All right. Now, what is going to be different in every protein is simply, in every amino acid, is simply going to be this R. Is going to be the one changing. Every other ones remain constant. You're going to have a carbon, you're going to have a carboxyl group and the amino group right there. Now, here are the amino acid that we have listed here. There are 20 amino acid that we talk about predominantly. So you see the H here is the difference here. Every other thing is the same. Here it's CH3 in alanine. In glycine, it's just H. In valine, it has become this. In leucine, it has become this. In isoleucine, it's now this. In proline, you see this. In tryptophan, is this. In phenylalanine, is this. In methionine, is this. Serine, these are switched to HIF. And now you have that. Now, this is non-polar side chain hydrophobic. This is polar side chain hydrophilic. And these are acidic, negatively charged amino acid. These are positively charged amino acid. Now, polypeptide, amino acid, I told you already that the bond that holds amino acids together are called the peptide bond, all right? And that is what you see there. Each bond that joins an amino acid is called peptide bond. Again, the same thing, monomer plus monomer produce polymer, and that is produced through dehydration reaction. Anytime a new bond forms, most of the time it is dehydration reaction joining smaller molecules to form a large molecule. All right. Now, when you look at this, like I told you earlier, a polypeptide, I mean, a protein that is functional must be a three-dimensional shape protein. And again, how did it become three-dimensional? It has been twisted, folded, coiled into a shape that is unique, which is what you see here. All right, let's go. Now, I'm going to talk about the different shapes of protein. Like you are seeing there again, that proteins are only functional at a three-dimensional shape, which is what you see here, three-dimensional shape. Now, we have four types of protein. We have the primary protein, secondary protein, tertiary protein, and the quaternary protein. All right. Now, the primary protein is simply a sequence of amino acid. It's just a straight chain of amino acid. It is not going to perform the functions that we highlighted about protein because it is not three-dimensional yet. It's just straight line. The second kind of protein, which are the secondary protein, they are getting to be coiled. They are not three-dimensional shape yet. So they are still not going to perform the function of protein in transportation, cell communication, cell identification, all of that. They are not going to perform it, but they are building block for protein, right? Yes. So we call them alpha helix and beta sheet. You are going to see that as we go. So here is just a straight chain protein. But before I come here, then we have the third one, which is when it's becoming three-dimensional shape now. And the fourth one is also three-dimensional shape. So the tertiary protein and the quaternary protein are functional. So here you see this protein guy here. This is a primary protein. Why? Because it's just straight chain. It's just straight chain. It's not coiled. It has a beginning. It has an end. Now, you see this? This is a primary structure of protein. It's straight chain. Nothing else. Now, this is secondary structure of protein. Now you can see it is becoming an helical shape. And the second one you see here is a pleated shape, which is also, now when you look at this, you see there are bonds here. There are bonds all around here connecting this protein together. You see this amino acid, you see there are bonds here. This is making it to become coil, but it's still not three-dimensional shape. Now, another kind of secondary protein is still this beta pleat. All right, it's pleated. The first one is alpha helix. This is pleated. Now you see here, these are bonds connecting polypeptide chains together to become pleated. And now at this point, they have become coiled, folded, and twisted on each other. And now this is a tertiary structure that is also functional as protein, and then the quaternary structure. All right. So that is what you see. That's the bond and the self attraction that comes upon them. This is collagen. Now, this is the protein hemoglobin that carries oxygen for you to get oxygen supply in your blood. All right. Now you see it is a four-dimensional protein. It's a 4-unit protein, and the shape determines the function. That's why if anything happens to this shape, the protein will not be able to carry enough oxygen, which is what we call sickle cell anemia. Anemia simply means deficiency of oxygen, which is what you notice here. All right, so sickle cell is when there is a substitution that affects the protein shape, which will invariably affect the structure of the protein. When anything happens and the structure of protein is affected, we call it denaturation. So denaturation is a process. It could be high temperature. That's why when temperature rises beyond 104, they ask you to run to emergency, because high temperature, high pH can affect the shape of a protein and make a protein to lose its nature. At the moment a protein loses nature, we say that that protein has been denatured. Right? So I'm going to talk about nucleic acid in the next video. I need to get to class now. See you soon.